So in the last section, we talked about how the cerebellar system manages the sequence of behaviors that can support something like episodic memory retrievals. So now let's have a quick look at how those cerebellar processes are supported at a somewhat more detailed level. And the anatomical structure called a cerebellar microsome is the substrate for recording behavioral sequences, like the episodic memory retrieval that we've been talking about. A cerebellar microsome contains an array of cerebellar nuclei neurons. A different neuron, or small group of neurons, corresponds with each behavior in the sequence. And in the case of episodic memory retrievals, these neurons target the thalamus and establish synapses onto the thalamic neurons that fire when the corresponding behavior in the sequence occurs. The cerebellar nuclei neurons are inhibited by Purkinje cells in the cerebellar cortex. One Purkinje corresponds with one behavior in the sequence, and it inhibits all the cerebellar nuclei neurons except the one corresponding with its behavior. Hence, when the Purkinje fires, it allows just the cerebellar nucleus neuron corresponding with its behavior to fire, implementing that behavior through the thalamus. There's also a group of inferior olive neurons making up part of the microsome. One of these neurons targets just one Purkinje, and when the inferior olive neuron fires, its effect is to reduce the strength of all the recently active synapses on the Purkinje that are derived from the cortex via the pontine nuclei and the granule cells. The inferior olive neurons are inhibited by cerebellar nuclei neurons. If the timing of the behavior is incorrect, the inferior olive fires, changing the Purkinje receptive field to reduce the effect of recently active inputs. So the inferior olive tunes the timing of the behaviors in the sequence. So once again, we've mapped an understanding of how a sequence of cortical information release behaviors implements recalls of events, through the high-level anatomy to more detailed levels in the cerebellum.